Fabian Finger, who's here from the University of Copenhagen.
Yeah, that's true. Oh, thanks a lot, Morten. Yeah, I'm indeed a senior postdoc located here in Copenhagen in the group of Zach-Garrett-Heinz Center for Basic Metabolic Research. And what I'd like to do in the next couple of minutes is to actually guide you through the actual experimental work and the results that hopefully justifies that broad title that I choose here. In fact, I could have chosen this title too, because what we did actually is studying mitochondrial myopathy after cardiolipin loss, and then assessed physiological parameter both on the systemic but also on the tissue-specific level. So yeah, as you can see, well, our continued effort is like circling around cardiolipin. We yesterday heard like a little bit about it. Just to remind you, cardiolipin itself is a very unique phospholipid, predominantly located in the inner mitochondrial membrane and virtually associated to like all mitochondrial functionality. So it's not surprising that disrupting cardiolipin biology has then detrimental effects associated to a variety of pathologies and diseases as well as aging. For the rest of the talk, please keep in mind that cardiolipin is synthesized de novo, and the key enzyme here is called cardiolipin synthase 1, or short CLS1. So, as I said, we are generally interested in tissue-specific effects of cardiolipin. Yeah, in this project, specifically skeletal muscle. And there's the interesting notion, which is probably not only true for skeletal muscle, but more in general, that cardiolipin levels seem to drop with age. Yeah, it's, in my opinion, insufficiently shown, but this is what's out there. Still, we were asking whether this is a consequence or cause. Historically, this is seen more as a consequence, as a bystander effect of decreasing mitochondrial mass. However, the reason why we went ahead is actually from our observations, where we took young and old mice and then checked the expression level for biosynthesis factors for cardiolipin, and we saw that the most significantly downregulated one is actually CLS1. So we have a transcriptional response associated to age, which then also translating into lower cardiolipin level as proposed in the literature. Interestingly, this is also reflected in kind of a similar way when we look into young versus like elderly humans. So this all was justification enough for us to actually dig deeper and see whether cardiolipin loss itself can drive certain features of skeletal muscle aging. So we did that, but also paid extra attention in a sense that we compared different muscle types. And if we choose the soleus and EDL as representative of different muscle types, just because they are the most extreme with respect to metabolic, inherent metabolism and contractile features, namely soleus being slow-twitched and predominantly oxidative, EDL, glycolytic, and fast-twitch. So how do you study that? Yeah, you create a genetic mouse model where we tissue skeletal muscle, specifically knock down CLS1. As you can see here, it works as predicted. Transcription levels for CLS1 go down only in skeletal muscle, no other tissues involved, which translates into dramatic loss of cardiolipin in skeletal muscle. How do these mice look like? They're smaller, so they're lighter. Their body weight is lower in comparison to the control, which is solely attributed to the inability to gain lean mass. Fat mass is unaffected in these mice. And when you look closer, like on the tissue level, you will hopefully appreciate that we have an undisproportional, decrease in muscle mass, specifically in glycolytic muscle, which is in itself actually seen in aging and is part of like an aging hallmark. So we went ahead and characterized the mice deeper. As I said, cardiolipin is quite important for all mitochondrial functionality, so we assessed their respiratory capacity. We isolated single fibers, permeabilized them, and ran them through the Ouroboros system. And what you can see is, and this is not really surprising, that oxidative muscle actually show decreased respiratory capacity. However, the surprise comes when you look into glycolytic muscles, because respiratory capacity is absolutely maintained and indistinguishable from controls, even though we don't have cardiolipin anymore. And also the mitochondrial architecture, especially the crystal architecture, is indistinguishable between oxidative and glycolytic muscle. And as you can see and hopefully appreciate, mitochondria are not very well off under cardiolipin loss, but still can respirate. Another type of characterization is obviously to look into the fiber type composition. In mouse, there are 4 different ones. So we stained muscle samples of controls and cardiolipin deficiency model. So in blue, you see type 1 fibers representing the most oxidative ones. In red, you see type 2b fibers, most glycolytic ones. And also the fast twitch, blue being oxidative and slow twitch. When you quantify these stainings, you can see on the lower left that we also here have a disproportional decrease in the MHC stain for glycolytic fast twitch type 2b muscle fibers, which is actually due to the fact that the cross-sectional area of those fibers actually decreases in our model in comparison to the controls, which again is considered a hallmark of skeletal muscle aging. So now going into the more metabolic phenotyping here, we first looked into fatty acid oxidation, which is prevalent in oxidative muscle, which is like the feature of oxidative muscle. And on the left side, you see a graphical depiction of proteomics data highlighting that, well, it's always the left box is the soleus, the right box is the EDO. And you can hopefully appreciate that there's an upregulation of factors involved in fatty acid oxidation in both muscles, but especially in glycolytic muscles. And to get a feeling for whether there is more activity and really more oxidation, we can measure enzyme activity of HAT. And to our surprise, we don't see any changes in oxidative, but we see massive changes in glycolytic ones. So essentially, glycolytic muscle tries to become an oxidative muscle, quite successfully actually, because it ramps up the capacity to oxidize fatty acids almost to the level of a stereotypical oxidative muscle. Also, that's considered an aging hallmark. So we went how we were interested. I was like, okay, we looked into fatty acid, but how does glucose behave? And there where it became like really surprising to us, because even though we have higher fatty acid oxidation, glucose uptake is through the roof across the board of skeletal muscle. So higher glucose uptake, which is so strong that it actually results in an effect on a glucose tolerance paradigm. Which is, I also want to mention, that is independent of effects on insulin sensitivity. So question is, nice, we have more glucose uptake. Where does the glucose go? So again, proteomics data here, just highlighting, which is what you see is the classical glycolysis pathway, where you see only one enzyme upregulated, which is hexokinase 2. Makes total sense. Glucose is coming in, gets phosphorylated, and actually trapped inside the cell. So, but the question is, it doesn't look like, and I know this is just proteomics, but it doesn't look like it's going through the route of glycolysis. What happens instead is when we also then factor in snapshot metabolomics, which happens is that glucose is shunted away from glycolysis into pathways like the pentose phosphate pathway and 1C metabolism. Interestingly, all these pathways are known to fuel antioxidant defense, which in our opinion makes a lot of sense because we also measure increased mitochondrial ROS in the absence of cardiolipin in skeletal muscle. So we have Apparently, an oxidative switch, a metabolic switch, towards fatty acid oxidation, still higher glucose uptake, which is used not for energy production, but for antioxidant defense. This goes hand in hand with an increase of virtually all oxidative stress markers. Just want to highlight two of them for their popularity, and also the amplitude of the regulation here. So you see FGF21 expression and GDF15, well, FGF21 expression and serum levels, and the same for GDF15, high across all skeletal muscle, indicated of strong oxidative stress, and actually resulting in a tremendous increase of both of these factors in the serum. Interestingly, when we use an inducible knockout model, where we, through tamoxifen, can actually knock out cardiolipin synthase in adult mice, you can hopefully appreciate that for at least, and that's where we ran the time, where we stopped the time course, for at least the first 2 months after getting rid of cardiolipin synthase, the increase of FGF21 and GDF15 is solely attributable to glycolytic muscle, highlighting the fact that it indeed makes sense to investigate both extremes of skeletal muscle with respect to the metabolism, and also highlighting the fact that glycolytic muscles are indeed more sensitive to age-associated oxidative damage. So as a pre-summary, and I think I mentioned that often enough, but all of what I just showed you is considered to be an aging hallmark, which actually makes, for skeletal muscle, which makes us actually confident that we can use cardiolipin synthase or cardiolipin deficiency in skeletal muscle as a tissue-specific accelerated aging model, which obviously then opens up a lot of interesting questions of future projects, probably. The second summary is more mechanistically, because what we were able to, by just directly comparing glycolytic and oxidative muscle under the paradigm of cardiolipin loss, and factor in metabolomic data and metabolism assessment, is basically coming up with a concept describing a global antioxidant response based on the actual inherent metabolism. And this is, in our mind, is a useful umbrella term, and it's short gamma. So I want to stress again that the disappearance in this model of glycolytic muscles, and the decline of glycolytic muscle in this model, solely depends on the inherent metabolic makeup. And the fact that glycolytic metabolism is not suitable to fight oxidative stress efficiently. And this is why actually glycolytic muscle almost changes its entire identity, both on the contractile and on the metabolic level. And this is then also, when I come back to my initial title, this is the same answer in my mind for aging as well. If we take into account that cardiolipin deficiency actually mimics lots of those aging hallmarks in skeletal muscle, the answer remains the same. The answer to why glycolytic muscle declined first during aging is rooted in its inherent metabolism. And with that, I would like to stop, acknowledge a fantastic team of scientists. It's a huge list, 40+ people, funding sources, and of course, Zach as my PI and the entire team. And yeah, thanks for your attention.
Thank you so much. We're actually out of time, but really appreciate your contribution. Amazing. Please find Fabian after and ask about cardiolipin and slow and fast twitch fibers.